Genetics, Section 7, RNA and gene expression. In this section, we've got our bell ringer. We'll then look at key ideas. Major points of emphasis for this section are an overview of gene expression. We'll talk about RNA and what its job is. Transcription, how transcription reads the genes. We'll talk about the genetic code. So what are the three-letter words that make up the genetic code? Translation we'll talk about. And that's the process of taking RNA and creating proteins. And then we'll look at the complexities of gene expression and summarize the entire section. So our bell ringer. There's a number of different chemical structures that are involved in physiological processes. Examples include hemoglobin in the blood, insulin that helps to regulate the amount of glucose or the amount of sugar levels that we have inside of our bloodstream, and enzymes that regulate body functions. All of these things are made up of proteins. So your bell ringer is to name some other parts of the human body that contain proteins. Don't name just one or two. Give me 3, 4, or even 5 different parts of the body that are made up of proteins. Our key ideas. What is the process of gene expression? So how are genes expressed? What is the role of RNA and what is its job as far as gene expression? What happens during transcription? How do codons determine the sequence of amino acids that result after translation? So we'll really look at the difference between transcription and translation in this section. We'll look at the major steps of translation, and then do traits result from the expression of a single gene? So that's kind of one of our all-encompassing questions for this section. Alright, so overview of gene expression. So we've talked quite a bit about DNA and talked about how DNA is the information that cells use in order to make proteins. However, DNA is not directly responsible for creating proteins. Ribonucleic acid, or RNA, it's a separate type of nucleic acid. And that type of nucleic acid actually takes the information from DNA that's found inside of the nucleus, and it makes a copy of that DNA in order to eventually create the proteins. So gene expression is the manifestation of genes into specific traits. So it is taking the DNA and is taking those genes and it is making them into specific traits that would make up our bodies, whether it be brown hair, black hair, blue eyes, brown eyes, skin pigmentation. It could be a number of different expressions that our body physically has. So gene expression produces proteins by transcription and translation. This process, it takes place in 2 different stages, both of which involve the use of RNA. So the first stage of gene expression is called transcription. Transcription is when the information that is in the DNA is actually copied and a strand of RNA is made. So DNA inside the nucleus is copied into a strand of RNA. And transcription is similar to copying or transcribing notes from the board. So taking DNA, the board would be the DNA, and then putting that information in a notebook would be like RNA. Okay, so up on the board you might be taking notes off of the board. You can't really pick up the board and carry it along with you everywhere you go with your notes. So what you do is you make a smaller copy of certain parts of what's on the board into a notebook. So DNA would be the board, the notebook would be RNA. The second stage of gene expression is translation. Translation takes the information that has been copied into RNA or made into RNA. That strand of RNA leaves the nucleus. Usually this occurs on the ribosome, so that section of RNA leaves the nucleus, goes out to the ribosome, where proteins are assembled based on the codes in that strand of RNA. So translation is similar to translating a sentence in one language into another language. So in this case it would be the language of DNA is copied into RNA, And then that RNA is then translated even further into specific proteins or amino acids. So again, transcription, you'll notice right here inside of this cell, we've got our DNA, which is housed inside the nucleus. Transcription is going to occur when a strand of RNA is made from the DNA. Later on, this RNA leaves the nuclear membrane. It goes through the nuclear pores. It comes out into the cytoplasm of the cell where it binds with ribosomes. And this section of RNA is translated into specific proteins. And there's a number of different enzymes that help this process to occur. These enzymes will bind on the strand of RNA, and based on the sequence of nucleic the bases, nitrogenous bases, based on their sequence, it will create an overall code for what proteins are created. So let's specifically look at RNA. All of the steps in gene expression involve RNA, and there's a number of different types of RNA that occur inside of our body. There's specifically 3 types of RNA that help to complement DNA and help to translate the genetic code into proteins. RNA is made up of nucleotide subunits, much like DNA. So we talked about adenine, thymine, cytosine, and guanine in DNA. In RNA, most of those are the same with the exception of one. So RNA differs from DNA in 3 ways. First, RNA usually is composed of 1 strand of nucleotides rather than 2 strands of nucleotides. Whereas if we remember, DNA had 2 strands of nucleotides that were bound together by some weak hydrogen bonds. In order for that DNA to be copied or replicated, those hydrogen bonds would have to be broken. Second, the RNA nucleotides contain the 5-carbon sugar ribose rather than the sugar deoxyribose, so hence the name RNA instead of DNA. So it's just simply the 5-carbon sugar ribose. And then third, RNA nucleotides have a nitrogenous base change. Uracil occurs instead of thymine. So in the place of thymine, RNA always has uracil. Uracil, again, much like thymine, it's complementary to adenine. So whenever RNA pairs with another nucleic acid, it is always going to have that uracil, and that uracil, again, takes the place of the thymine.
The instructions in DNA are carried out with the help of RNA. Here you can see a DNA molecule and an RNA molecule. Notice that RNA differs from DNA in 3 ways. First, an RNA molecule has only 1 strand of nucleotides. Second, RNA contains ribose instead of deoxyribose. Third, RNA has a nitrogen base uracil instead of thymine.
The 3 main types of RNA that we find inside the human body include messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. Messenger RNA, again, messenger RNA is the one that is produced inside of the nucleus when DNA is transcribed into RNA. So then that messenger RNA carries the instructions for making a protein from a gene and delivers the instructions to the site of translation. And in this case, a lot of times the site of translation, again, is the ribosome inside of the cell cytoplasm. Once that mRNA reaches the site of translation, transfer RNA then reads the instructions, and it translates the mRNA sequence into protein subunits. These subunits are amino acids. So it looks at the mRNA sequence, and it copies that mRNA sequence into amino acid sequences. Ribosomal RNA, or rRNA, it's an RNA molecule that is part of the structure of ribosomes. So it helps to make up the structure of the ribosome where this protein creation or protein synthesis is occurring. Ribosomes are the cellular structures where protein production occurs. Please remember that there are numerous ribosomes found within a cell, most of which float around in the cytoplasm. They're very small. Some of them can actually attach to the ER, the endoplasmic reticulum, and at the site of the endoplasmic reticulum where they are attached, it actually creates kind of a bumpy outer surface to the ER, which in that case we call that ER rough ER.
RNA exists in 3 different types. Each type of RNA has a specific function. Messenger RNA, or mRNA, consists of a single uncoiled strand of RNA nucleotides. mRNA acts as a messenger between DNA and the ribosomes, carrying the information from a single gene to be translated into a protein. Transfer RNA, or tRNA, consists of a single strand of about 80 RNA nucleotides folded into a cloverleaf shape. Each type of tRNA binds to a specific amino acid. One end of the tRNA molecule contains an anticodon, which binds to the complementary codon on an mRNA molecule during protein synthesis. Ribosomal RNA, or rRNA, binds to several proteins to form ribosomes. rRNA interacts with mRNA and tRNA to direct protein synthesis.
So going back and talking more about transcription, this is the point in which the gene is actually read. So remember, each one of your genes are found on chromosomes, and again, chromosomes are made up of these long coiled strands of DNA. So anytime we're reading a gene, we're reading DNA. So in transcription, again, the information of the gene, the DNA, is transcribed or it's copied into mRNA. For this process to occur, for transcription to occur, there has to be a protein called RNA polymerase. And RNA polymerase attaches to the DNA strand. As it attaches to the DNA strand, it slowly unzips the DNA, and it ends up causing that strand of DNA to slowly make a copy of each one of the genes. A specific DNA sequence in the gene that is that goes through this process is called a promoter. So transcription begins when RNA polymerase binds to the specific DNA sequence in the gene, and again, that site, that actual part of the gene that allows RNA polymerase to bind is called a promoter. RNA polymerase then unwinds and separates the 2 strands of the double helix to expose the DNA bases on each strand. So RNA polymerase, it basically attaches to the DNA. As it attaches to the DNA strand, it slowly adds complementary RNA bases as it reads the DNA, as it reads the genes. So as RNA polymerase moves down the strand of DNA, a single strand of mRNA grows off of that RNA polymerase. Behind the moving RNA polymerase, the 2 strands of DNA close up and they reform the double helix. So the RNA polymerase does both the jobs of opening up the DNA to make copies of the make copies and change it into mRNA, and it also closes it back up at the opposite end.
At the beginning of transcription, the DNA containing the gene of interest unwinds, forming a template strand and a non-template strand. Only the template strand is used to synthesize RNA. An enzyme called RNA polymerase adds complementary RNA nucleotides to the newly forming RNA molecule. When the RNA polymerase reaches a DNA region called a termination signal, it releases the DNA strand and the RNA molecule.
So it's important to remember that your genes are only created off of the template strand of DNA. So that template strand is the strand that RNA polymerase is using to create. So transcription is not the same process as replication. Don't make that mistake. Replication is when the DNA is copied for new cells. So we go through that process of the cell cycle where a cell, it grows, it divides, mitosis occurs where the nucleus is copied. That process is completely separate from this process of transcription. Okay, so DNA replication is when a copy of DNA is made. Transcription is when we make mRNA from the strand of DNA. So in transcription, a new molecule of RNA is made from the DNA. In DNA replication, a new molecule of DNA is made from the DNA. Alright, so this picture further explains that process of transcription. Number one, RNA polymerase binds to the gene's promoter. Remember, the promoter is the section of the gene that says, okay, attach here and start making mRNA. So the RNA polymerase attaches. Step two, the two DNA strands unwind and they separate. And then step three, complementary RNA nucleotides are added. So over here in step three, the RNA polymerase is adding new nucleotide sequences again to the template side of DNA. So we're looking for the template side of DNA, and it's slowly creating this big long chain of mRNA. That mRNA then can leave the nucleus where it can attach to the ribosome in the cytoplasm, and this process of translation can continue and create the proteins that make up our bodies. So the genetic code, and this genetic code is based on three-letter words. So a three nucleotide sequence is called a codon. So what happens is we end up with this section of mRNA. And this section of mRNA has, for every three nucleotide sequences, it has what's called a codon. And each codon corresponds to one of 20 amino acids. Remember, amino acids are the building blocks that make proteins. So for every three nucleotide sequence, a certain amino acid is attached. Those amino acids, as they form long strains, they eventually build proteins. Codons act as a start or stop signal for translation. So they tell where translation will start and where translation will stop. There are 64 mRNA codons, so there's 64 different sequences, different three-letter sequences that code for this process. Each codon specifies only one amino acid, but several amino acids have more than one codon. And we'll see that in the next chart that we look at here in a little bit. We'll see how a number of different codons can actually, or a number of different amino acids can actually have more than one codon. And this system of matching codons and amino acids is called the genetic code. And we talked a little bit about how we've deciphered the human genome. And part of this process was trying to figure out how do these different codons, how do these different arrangements of nucleotides, how do they create the amino acids that eventually are put together and create our proteins. So the genetic code is based on codons that each represent a specific amino acid. So number one, first we find the first base of the mRNA codon in the column of the table. So we find the first base right here. So if we had a section of mRNA that starts with U, we would look in this entire row right here. So we start there. If it started with a C, we would look through this row. A, we'd start with this row. G, we'd look through this row. 2, we would follow that row to the column that matches the second base of the codon. So if I said that you had a section, a codon that was UCG, we would start with a U for the first base. Then we would go over to the second base and we would see where those two intersect. And we would look in here and we'd say UCG codes for the amino acid serine. So finally, we would look for the third base over here, where we'd move up and down in that box until we match the third base. So we would go U, C here, G across. Again, same answer. Looking more specifically at this process of translation, this is the process of taking the RNA and copying it into proteins again. Translation occurs in a sequence of steps where it involves 3 kinds of RNA, and it results in a complete polypeptide. Translation takes place in the cytoplasm, where transfer RNA, ribosomal RNA, and messenger RNA all interact to assemble proteins. A specific amino acid is added to one end of each transfer RNA. The other end of the transfer RNA has an anticodon, and an anticodon is a 3-letter nucleotide sequence on transfer RNA that is complementary to the mRNA codon. So transfer RNA anticodons, they attach to the mRNA codons. They're basically like a jigsaw puzzle, where we've got the transfer RNA anticodon based on its sequence will match the mRNA codon. So the mRNA joins with a ribosome and transfer RNA. A transfer RNA molecule that has one correct anticodon and amino acid binds to the second codon on the mRNA. And peptide bonds form between the 2 amino acids. Basically what happens is the 2 new sections of amino acid just bind together. And the first transfer RNA is released from the ribosome. The ribosome then moves one codon down the mRNA, where the next section can be read, a new anticodon can come and attach, and it can create longer and longer strands of amino acids. So this video represents this process.
Ribosomes are the factories where proteins are made. Ribosomes are made up of 2 parts, a large subunit and a small subunit. These ribosomes attach to a copy of the DNA called mRNA. Together, the large subunit and small subunit form 2 grooves, called the P site and the A site. Transfer molecules, called tRNA, fit into the grooves. Each transfer molecule recognizes a specific code on the mRNA molecule and carries a specific amino acid. As the ribosome moves along the mRNA molecule, the P site holds the growing protein chain, and the A site takes in new transfer molecules with amino acids.
And so the longer and longer this ribosome moves along this messenger RNA, the longer and longer the chain of protein becomes, the longer the chain of amino acids. So these 2 amino acids would bind with this amino acid, this transfer RNA would fall off, the A site transfer RNA would slide over into the P site, and a new one would attach. And this process would happen over and over again, where longer and longer strands of amino acids would be built. So again, the amino acid chain continues to grow as more and more of that transfer RNA is attached, and it releases its amino acid that it's carrying, and peptide bonds bind those amino acids together. That transfer RNA, each time it loses its amino acid, it's just simply released. So the process is repeated over and over and over again until on the messenger RNA there is one of 3 stop codons that are reached. A stop codon is just simply a site in the mRNA that says stop production of protein. Many copies of the same protein can be made rapidly from a single mRNA molecule because several ribosomes can translate the same mRNA at the same time. So what will end up happening is they'll attach in numerous locations along the mRNA, and this chain of proteins will build a lot quicker. Basically the saying, many hands make light work, that's basically the process that's occurring when many ribosomes attach to the same copy of mRNA.
The messenger RNA sequence, AUG, signals the beginning of a protein chain. A transfer RNA molecule carrying a modified form of the amino acid methionine binds to the start codon, along with the 2 subunits of the ribosome, to begin translation.
So remember, AUG always represents a start codon. So this AUG on the mRNA is saying to the transfer RNA, this is the point where we're going to start production of proteins.
The ribosome reads along the mRNA molecule in groups of 3 nucleotides called codons. Each codon codes for a specific amino acid. When the ribosome reaches the sequence UUA, UAG, or UGA, translation stops. These sequences are called stop codons.
So going through this process of translation again, step number 1, the ribosome, mRNA and tRNA, which is carrying the amino acid, they bind together. Step 2, a new transfer RNA arrives and binds to the next codon on the mRNA. A peptide bond forms between the first amino acid and the amino acid created by this transfer RNA. So a peptide bond occurs right here. So it slowly starts creating this long chain of amino acids. Step 3, the first transfer RNA detaches and it leaves its amino acid. With each new transfer RNA, the amino acid chain grows. So it gets longer and longer and longer. Step 4, the process ends when a stop codon is reached. So in this case, UAG, right here, is the stop codon. So it's saying stop the production of proteins or stop the transfer RNA from binding. So in this case, this big long chain of amino acids just falls off and the process of creating that protein is stopped. Step 5, the amino acid chain is released and the ribosome complex falls off. So this process of gene expression is much more complex than just what we saw right here. The relationship between genes and their effects is very, very complex despite the neatness of the genetic code. So every gene cannot simply be linked to a single outcome. Some genes are expressed only at certain times or under specific conditions or specific environments. For example, at the beginning of one of these sections we talked about how the Arctic fox creates a winter coat and it also has a summer coat. So that's an instance where genes are expressed under certain times or certain conditions. In that case, it's an environmental factor that causes certain genes to turn on and or turn off. Variations and mistakes can also occur within our genetic code. So this leads to some complexity and it leads to some even mutations that can occur that can be either negative or I suppose in some cases maybe possibly positive. It is important to note that we have yet to see a positive mutation in any organism. The final outcome of gene expression is affected by the environment of the cells, the presence of other cells, and the timing of gene expression. So in summary, gene expression produces proteins by transcription and translation. The process takes place in two different stages, both of which involve RNA. In cells, 3 types of RNA complement DNA and translate the genetic code into proteins. During transcription, the information in a specific region of DNA, a gene, it's transcribed or copied into mRNA. And remember that that process occurs inside of the nucleus of the cell. The genetic code is based on codons that each represent a specific amino acid. Translation occurs in a sequence of steps, and this occurrence involves 3 kinds of RNA, and it results in a complete polypeptide. The relationship between genes and their effects is complex. Despite the neatness of the genetic code, every gene cannot be simply linked to a single outcome. So it's important to realize that yes, this process is very neat. Yes, it is meant to keep things from happening inside of your genetic code as far as mistakes go, but it is not flawless. There are mistakes that can occur, and in that case we have to have these built-in barriers that either destroy the cell that has had copies that are incorrect made, or there has to be some other way to go back and fix the mistake within the genetic code. And so our body does have some built-in mechanisms for helping that to occur.